No, I didn't. And you can. Yeah, sorry. Oh, no, I. Oh, shit. I know that. Oh, yes. Hello. Well, I thought. I was like, we're not. There's. Right. I've been like.
I'll get us started, I think, because sometimes they're classy. Afterwards, we still got people coming in, which is no surprise. Awesome crowd. Thanks for coming. My name's Denver. I've had the pleasure of serving as Emily Taylor's basketball. Where's Gia? Okay. All right, so I think we're good now. Thanks everybody. So I'm Denver. I serve as Emily Taylor's master's advisor, which has been a huge honor the last 3 years. I'm going to say a few words. I guess I got to set my coffee down now that I have a mic. So
Thanks for joining us. Really appreciate it. Appreciate the support. So Emily got her bachelor's degree from Iowa State University, where she did some really great work on plant parasitic nematodes, and then joined us 3 years ago or so as a master's student. Okay, and she has done just so much in that time, and has been really a core, important piece of what we're trying to do with the team. So you'll hear a lot here, and I'm not going to do spoilers to the best of my ability, but some things you might not hear about. Are that she really helped me develop and deliver a new difference, power, and oppression course. So I'd been teaching it for some time at the 100 level. We had to make it 300 level. She really helped me out with that process and has been teaching it with me the last few years. Another super important thing, supporting each other. In the IB community, we had crisis a year ago, a big tragedy. And I knew I was going to have to do a lot. I was department head back then, and I knew I didn't have to worry about teaching because Emily was so awesome. She covered it. So that's so important during that time. Emily supported a lot. She also, understanding how we bring these topics into our space, helped me develop a workshop with the Difference, Power, Depression leadership about how we bring science theories and social justice theories together and talk about those in the same spaces. Let's see here. She also helped me develop and deliver transdisciplinary research in Nepal and run a study abroad program in Nepal, which was just amazing. And it was really this last fall where I saw Emily just become this amazing leader. Just so cool, so amazing, and giving rise to what we see here today. The last bit, you will hear about some research here, a couple chapters that are just amazing. Both of them are very close to publication. We've gotten feedback from co-authors on one of them, and it'll be, kind of in the next month or so after this, they'll both be submitted. So last, I just want to say, Emily really embodies the values of our research team, transdisciplinarity, inclusivity, and compassion. And from there, I'm going to pass it to Emily.
Thank you so much, Dee. Can everyone hear me all right? Okay. So thank you so much, everyone, for being here today. My name is Emily Taylor, and I'm going to be presenting the culmination of my master's thesis research, titled Experimental and Ethical Exploration of Nematode Infection in Banana Slugs. So I just wanted to give a roadmap of the talk today. I'll be starting with an overall introduction, and my thesis consists of 2 chapters. So we'll move through chapters 1 and 2, ending with an overall conclusion and then into some acknowledgements. Okay, so I want to start today by talking about biodiversity. So biodiversity we know to be extremely important to ecosystem health and stability. And when you think about the word biodiversity, especially in conjunction with conservation, or maybe thinking about the term endangered species or extinction, we might think of some of these examples up here. Tigers, red pandas, maybe a tropical reef ecosystem or tropical birds. And these are all very charismatic or maybe larger species. So it might surprise you if I say that 38% of all documented known animal species extinctions since 1500 CE are actually mollusks. And mollusks meaning soft-bodied organisms that are invertebrates, and they exist in damp or aquatic habitats. And even more, let me move the mouse. Even more so, on top of that, 90% of all of those mollusk extinctions are coming from terrestrial gastropods, or slugs and snails that live on the land. And so that is the group that I'll be focusing on today. So here in Oregon, we actually have a very rich biodiversity of terrestrial gastropods. We have some amazing species here, some species that can only be found in the state of Oregon and nowhere else in the world. And so this is a really important area of biodiversity that we really want to protect. And so understanding some of the threats to this group are important. Most of them are anthropogenic in nature, and that includes habitat loss, climate change, pollution, as well as introduced species. And that is one that I'm going to be focusing on today, specifically in the context of biological control, also known as biocontrol. And biocontrol just means using another living organism that acts as a natural enemy of a pest that you want to target in order to control it without using something like chemical pesticides. And a really good example you might think of for this is using ladybugs in your garden to control aphid populations. And so in thinking about introduced species and controlling pests, there's one pest that I want to highlight, which is the gray field slug, Duroceras reticulatum. And this little tiny slug is extremely destructive all over the world in agricultural crops, but is also really impactful here in Oregon as well, in the Pacific Northwest more broadly. Specifically in Oregon, the gray field slug causes $60 million of damage every year to the grass seed industry alone. So this is a huge pest and a huge problem that we have in this area. And so in thinking about a possible biocontrol approach for controlling this issue, we have the opportunity to explore Pelioiditis nematodes. These are a genus of nematodes being small roundworms that typically live in the soil and are sort of microscopic. But the Pelioiditis genus of these nematodes associate with terrestrial gastropods, and they're facultative parasites, so they can live on a slug or snail as a host, or they can be free living, feeding off of bacteria. And there are a couple of European strains of these nematodes that have been commercialized and sold as an actual biocontrol product called Nemaslug. Usually the main target is that little gray field slug that I mentioned before, Duroceras reticulatum. And as I said, is the most crop damaging pest slug in the Pacific Northwest region of the US. So up here you can actually see an image of a gray field slug and there's a nematode inside of it. So the natural enemy system that this product is taking advantage of is the life cycle I have depicted here, where Pelioiditis nematodes have a third stage infective juvenile part of their life cycle, where they'll go and seek a gastropod host, enter through natural openings, and get inside and start to infect and sort of deteriorate the organism from the inside out. Eventually, the slug will die, and you can see this image. All of those fuzzy things are thousands of nematodes that are feeding off of the corpse and will reproduce and move back into the soil in search of a new host. So these are some images of what the Nemaslug product actually looks like. The nematodes themselves are sort of dried down into a powder, and you mix it with water to apply to your crop or to your garden. And there are two different formulations on the market. We have Nemaslug 1.0, which is made up of Pelioiditis hermaphrodita nematodes, which I will abbreviate to PH in the presentation. And secondly, we have Nemaslug 2.0, a newer formulation that is using the species Pelioiditis californica, or PC nematodes for short. And so this product is sold in other places of the world, in Canada, Kenya, the UK, and the EU, but it's currently not permitted for sale in the US, and that is for a couple of reasons. The first one is that it's unknown if the Pelioiditis nematodes are originating in the US, so whether they would be an introduced species that could potentially be a threat to our local biodiversity here. And secondly, it's unknown if there are non-target effects on organisms that are not, or species that are not meant to be controlled as a pest population. And so that brings me to my non-target focus of today. You may have seen around here is the Pacific banana slug. So this is a very large and well-known slug. The scientific name is Aerolimax columbianus. They play a really important role in the ecosystem as detritivores, decomposers. They play important roles in terms of seed dispersal, as well as just being a prey item on the food web. And they also have a lot of pop cultural relevance. You might have seen them as the UCSC mascot, this anthropomorphic slug, or on Vince Vega's shirt. Or in the beginning of Coraline, you might have noticed a banana slug, or maybe you even saw this guy at Powell's before. So clearly they have importance, and we totally recognize and care about this slug. And this is not a target of the Nemaslug product, and something that we want to protect, as well as the other larger scale biodiversity in this area. So that brings me to my Chapter 1 research, which is titled 2 Pelioiditis Biocontrol Nematode Species Infect Aerolimax columbianus, the Pacific banana slug, and increase mortality in laboratory infectivity trials. And so my objectives for running this experiment were to, 1, determine whether the biocontrol nematodes PH and PC cause mortality or death in a native and non-target species, A. columbianus, the Pacific banana slug. And secondly, I wanted to investigate whether there are differential effects between 2 different life stages of the banana slug, those being juveniles and adults. So my main question here is, is this product that it is intended for an agricultural pest going to impact a native species that we care about and we don't want to hurt, especially if this product is brought over to the US? And secondly, understanding if there is a difference between infection in juvenile organisms or adults is really important in trying to assess how this would actually come into contact with the ecosystem that we have right now. And so my methods for carrying out this study were to run a 38-day laboratory infectivity assay. And so that is essentially just bringing the nematodes and the slugs together into contact with each other and seeing what happens. And so I tested 2 different life stages, juvenile and adult banana slugs, and exposed them to 3 different treatment groups, that being the pH, PC nematodes, and then a control treatment where no nematodes were added. The design was 5 individuals by 5 replicates by those 3 treatments. And I wanted to note that nematodes were applied to the treatment groups at the Nemaslug recommended application rate, which is what you get off the back of the box of the product that I showed you. That is 30 infected juveniles per centimeter squared. And importantly, there are different container sizes used for the juveniles versus the adults due to their size difference. And I wanted to note that the Nemaslug, the nematodes that are applied are relative to that area. And then at the bottom here, I have a schematic of how the experiment was carried out. We started in 2 different places with slug field collection and nematode growth. So on the slug side, I have an image of Dee and I collecting slugs in a field of skunk cabbage. They love to hide in the flowers of the skunk cabbage, as you can see. All the slugs were collected from one location in Newport, Oregon. And afterwards, they were brought back into the lab and we weighed them. We determined which life stage group they were in based on that weight and different cutoffs. And then on the other side, back in the lab, we were also growing up a bunch of the pH and PC nematodes and then aliquoted them into their respective treatments in sterile water. And so bringing those 2 sides together on the first day of the experiment, I started the assay. And from then on, I monitored things daily. So I would check every slug for signs of infection and mortality, death, until day 38 of the trial in which it was ended. And from there on, I moved on to data analysis. So now I just wanted to show you what that actually looked like. On the left side here, we have an example of the juvenile container setup. In the middle is an adult container setup. You can see they have much different sizes. And then on the far right side, I am showing the incubator where all of the containers were kept. And so this is an incubator on a 12-hour photo period, and they were fed every 2 days. Okay, so I want to dive into the results, but firstly, I want to look at them qualitatively. So how did I see the infection occurring, and how did I know that it was the nematodes that were killing these slugs? So on the left-hand side, we have an example of a healthy juvenile taken in the field in Newport. And on the right is a juvenile showing the first signs of infection. So you'll notice that on the mantle, the head part, it's sort of constricted, and the whole body shape is a lot different than the one on the left. And if I zoom in on my microscope, I could actually see, oops, a little squigglies. Those are the nematodes actually on the surface of the slug. And eventually this individual died. And again, the fuzziness that you see on the body, those are all nematodes that are feeding off of the cadaver and reproducing as well. So infection in the adult group looked a little bit different. There's a healthy adult on the left in the field, and then on the right side, we have the first signs of infection, which actually looked like skin lesions in the adult group. You'll also notice just sort of like a misshapen body as well compared to the field image. And then eventually this adult passed away, and you can see really large lesions had spread across the body, a lot of discoloration, and in this individual, the tail end is actually dissolving into a bunch of nematodes. So switching gears to look at things more quantitatively now. What I'm showing you here is a simple bar chart of the raw count numbers of deaths by the end of the experiment. We're looking at 2 different time points with day 19, the halfway point of the experiment, and the lighter colors, and day 38, the end of the trial, in the darker colors. And so on the x-axis here, I have experimental groups. I color-coded them by treatment. So the orange ones are the pH, P. hermaphrodita nematode treatment. Blue is PC, P. californica, and the gray groups that you're seeing are the control groups. And so on the y-axis, we're looking at total number of slug deaths out of 25. I had 5 individuals by 5 replicates. So 25 out of 25 indicating complete mortality. And so I want to start by orienting on the control groups here. You can see that there was not that much death in the controls. We have 4 juveniles and 1 adult by day 38 of the experiment. And that is something that is sort of expected to see when you take organisms like this into the lab. That is striking compared to the pH treatment group. You see that both the juveniles and the adults reached complete mortality by day 38 of this experiment. 25 out of 25 individuals had died. And you'll also notice that in the juvenile group, they had already reached complete mortality by the halfway mark. And so next, moving to the PC treatment group, P. californica, we see that the juveniles had a lot of death, 20 out of 25 individuals, but they never reached complete mortality. And with the adults, there was very little death, only 2 by the end of the trial, which puts them, that group down there, similar to the levels of control deaths. And so diving in a bit further, I did something called Kaplan-Meier survival probability analysis of the banana slugs over time of the experiment. So here on the x-axis, we're looking at time in days, and on the y, we're looking at the calculated survival probability, with a value of 0 indicating complete mortality and a value of 1 indicating no mortality. So once again, I want to start by centering on the control groups. Those gray lines up there are the juvenile and adult controls with very little deaths. And interestingly, and reflecting what we saw on the last slide, the adult PC treatment group is also up there with the controls. And that is in contrast to the juvenile PC group, which experienced a lot of death, however, never reached complete mortality, but it is quite different than the adult group. And then next, looking at the P. hermaphrodita treatment groups, we see juvenile on the left, adult on the right. They both reached complete mortality, and it was about 10 days apart from each other. And interestingly, you might notice that both of the survival curve slopes are very similar to each other. So going a step further, I wanted to assess whether there were significant differences actually between these groups. So here I did a pairwise log rank test with a Bonferroni correction, and I wanted to highlight a few significant values of interest here. Firstly, I just wanted to know if the control groups were significantly different than the treatments. So adult pH was different from the adult controls. Adult PC was not significantly different from controls, which you might expect as they're grouped up there together on the graph. And then both juvenile treatment groups, PC and PH, were significantly different than the juvenile controlled group. Next, I wanted to look at comparisons between the life stage treatment groups. So in the juvenile PH, that was significantly different from the adults, and the same goes for the PC treatment. Juveniles and adults were different from each other. And then finally, just one interesting result here is that the adult PC and adult PH treatments were significantly different from each other, which is interesting given that they are both formulations of the Nemaslug product. So in conclusion, both Paleoditis nematode species significantly increased mortality in at least one life stage of the banana slug, with P. hermaphrodita infecting and increasing mortality in both life stages, juvenile and adult, whereas P. californica only did so in the juvenile life stage group. And so this is strong evidence to suggest that susceptibility may depend on host life stage. So just some overall takeaways from this research. I think that we should really use caution with Paleoditis biocontrol products. Clearly in this experiment, there was a non-target impact on a species that we really want to preserve the diversity of, especially here in the Pacific Northwest. And so I would also encourage really comprehensive host range testing to include multiple different species, different life stages, as well as settings. One limitation of this study is that it was done in a very controlled laboratory environment setting. And so understanding more about how organisms or how slugs might actually encounter these nematodes in the wild would be really helpful. On top of that, I just wanted to share some more personal takeaways that I had. I spent so much time with these slugs in the lab, just watching them, studying them, taking care of them. And it was really difficult to experiment on them, honestly. You saw the infection pictures. It was really brutal and kind of a slow decline process. So it was very sad. And I was reflecting on, I feel like there's a lack of discussion about this. As scientists, we're supposed to be very objective and not really talk about how things affect you like that. And then another thing that really struck me was that there were no required ethical concerns with my experiment, which is in stark contrast to some of the other research going on in our department where you have to go through a really regulated process. So I really went out in the field. I collected 180 banana slugs. No one even really knew that I did that except my lab, and I brought them back in and just subjected them to this experiment. So that was kind of striking to me, and it sort of got me thinking about what does animal research look like in the US in particular. So here what we have is something called the Institutional Animal Care and Use Committee, or IACUC. This is a federally mandated committee that protects all vertebrate animals used in research, teaching, or testing. And to get IACUC approval, this requires an inspection of the research facilities and a really regulated review process by a committee, wherein they'll do a cost-benefit analysis of, is your research worthwhile enough to justify using animals and having them suffer. And so there is actually only one mention of invertebrate animals. You'll notice they said vertebrates, and that does not capture what I researched on invertebrate terrestrial gastropods. And so the only mention in the handbook is that invertebrate animals such as the fruit fly have long been used in research and represent another type of living alternative to vertebrate animals. So I think this reflects a really interesting gap in our ethical considerations for testing on animals. There's sort of a distinction, a hard line between vertebrates and invertebrates, but it seems like that might not be fully encompassing of the ethical considerations that we could pay attention to. And others have thought this as well. Octopuses are also invertebrates, so they don't fall under this protection, but we understand them to be very intelligent and to even portray emotive states in some cases. So this is just a screenshot from Harvard Law, an article about people fighting to have more regulation over cephalopod research as well. So this is sort of a growing topic in the field. And in thinking about invertebrates, this just refers to animals that lack a spine. And they actually make up 95% of known animal species, an overwhelming majority if you look at this animal section of the pie chart here. And so it seems like a very arbitrary line to set in terms of ethical consideration, right? But we know them to be really great model organisms. They're easy to maintain and have usually really quick regeneration times. So you might think of the fruit fly, Drosophila melanogaster. It has been so instrumental in advancing our knowledge, especially in terms of human health research. Some examples include cancer drug development, diabetes research, as well as lifespan and aging. So it cannot be understated how important this type of testing has been to our understanding of human health systems as well. But that just sort of got me thinking about what are our ethical frameworks that we employ in modern science. And what I found is that most of our research, especially in terms of animal use in our research, comes from a utilitarian ethic. And so the idea there in utilitarianism is that the morally right action to take is one where the net benefit outweighs the net harm. So particularly in animal research, that's where the cost-benefit analysis comes in the IACUC approval process. There has to be an understanding of your research to be more beneficial than the amount of suffering that the animals would experience. So it's a very utilitarian system. And this is also the basis upon which the 3 R's of animal research stem from. This is a widely used framework in modern science. We have the 3 R's being replacement, using non-animal systems or those of lesser concern. Reduction is minimizing the number of animals used, and refinement is modifying procedures to minimize pain and distress. And so I think this is a really great place to start in thinking about animal research ethics, but people have brought up concerns like some of these might be ambiguous or maybe dependent upon the researcher or the parties who are doing this work. And sometimes one or more of the 3 R's can be in conflict with each other. So taking reduction and refinement, for instance, if you're refining a procedure to make it less painful or less invasive for a certain organism, that might require you to incorporate more animals into your research. So then the question becomes, is it better for more animals to suffer less or for less animals to suffer more? And so it's sort of, it's unclear sometimes in cases like this. And I think personally, the biggest loophole in this framework is that for some researchers, any level of knowledge that you can gain in an experiment is enough to justify virtually any level of animal suffering. So this is where the discussion of Buddhist ethics comes in. And so why Buddhist ethics? As you might know, in my lab, we do try to study the intersection of Buddhist philosophy and science. But even more than that, we are not the first ones to be doing this. There's a lot of discussion, especially in terms of animal ethics, but about taking Buddhist philosophical ideas and applying them to more modern concerns such as like climate change and just conservation. And so I have some examples of that here. Buddhist eco-philosophy is one of the newer terms, but just to show that this is a growing discussion in the field. And so I think this provides a useful comparison because it contrasts utilitarianism. It's really a different sort of framework. It also provides a non-Western perspective. And across all of this discussion, there are sort of 3 principles that have a lot of application to science and ecology. And the ones that I'm focusing on today are non-harm, interdependence, and compassion. And I do want to note that Buddhism is a very old religion that has many schools of thought and different lineages. And so I'll be drawing on these principles sort of in a foundational sense, not particular to any one school. So that brings me to chapter 2, which is the ethics of invertebrate animal research from a Buddhist philosophical perspective. And my objective here was to apply those principles of non-harm, interdependence, and compassion in order to evaluate non-human animal research ethics. And I want to use the chapter 1 experiment that I ran, the infectivity trial, as a case study to highlight alternative approaches to scientific inquiry. Okay, so the first one I'll start with is non-harm, or ahimsa in Sanskrit. This is the principle that one should not cause harm to others. And this is actually the first precept that a lay practitioner will take in practicing Buddhism. And so a quote from the Buddha is, "All tremble at violence, all fear death. Putting oneself in the place of another, one should not kill or cause another to kill." And so this also sort of has a logical basis to it. If you do not wish to be harmed, you should not enact that on any other living being. And while I will say this is practiced differently in different schools, it's important to note that in some cases, Buddhist philosophy has a understanding of context. So in my case for the experiment in chapter 1, my original intention and my goals for doing that were to potentially protect a larger group of organisms in the future, or maybe even other native species. And so that is relevant here. In some cases, Buddhist practitioners will still eat meat. So there's sort of a recognition of the context that matters. And so here I just have a little diagram. There's me as the scientist, the nematodes, and a banana slug. So in relation to the experiment, I definitely did cause harm to the banana slugs, and it was pretty visible. There's the juvenile that we saw earlier. There's an example of an adult, they had this like weird slang reaction to try and shed the nematodes off. And so this is certainly a case where the animals were suffering and they were harmed, and sort of a violation of this ethic. As for the nematodes, who can really say? It's a level of suffering that I'm not able to perceive. And so that also sort of reflects the biases of how we do conceptualize harm. And so the next principle I wanted to explore was interdependence, or pratitya samutpada in Sanskrit. This is the idea that all experiences and phenomena are deeply interconnected and that nothing exists in isolation. So oftentimes, like I said, the researcher is more objective and separate from their experiment, but this sort of framework would suggest that we are all interconnected. The banana slugs, the nematodes, and I were all existing in the same framework and influencing each other. So not only was I harming the banana slugs, but watching them suffer and seeing the result of that was also harmful to me. And I think that's important to acknowledge. And then finally, the next one I explored was compassion, or karuna in Sanskrit. This is the recognition of the suffering of all living beings, but even more than that, it's a commitment to alleviating this suffering. And so, in the context of this case study in particular, you might have noticed that in the adult PC group, not a lot of them died. And so there were a lot of leftover individuals that were alive at the end of the experiment. And they had been exposed to the nematodes, so I couldn't just release them, and I chose to put them in the autoclave and euthanize them at the end of the experiment. And I think that to understand compassion from this framework, it would have actually been my responsibility to take care of those slugs until the end of their life, despite the fact that they didn't die in the experiment. And that would have been more time, money, and resources for me personally, but I believe that under this framework, that was my responsibility that I had already taken on when I collected them from the field and brought them into the lab. And so I think that's a valuable piece that this framework can bring to the experiment. And the idea is that if we practice compassion to ourselves and to our research subjects, ultimately that will have larger impacts on our greater community and the world. So takeaways from this chapter were that the infectivity trial did reveal some ethical gaps. Looking at it from a different way, exposed some things that maybe could have been done differently, more ethically. Another thing was that suffering cannot be easily quantified. I feel like visibly I could see the suffering and discomfort of the organisms, but it's sort of hard when the frameworks that we do have don't have any guidelines for invertebrate animals. Also, researchers are not fully separated from their test subjects. I felt really heavily impacted by this work in a way that I really didn't expect. And so I think that's important to note. And then finally, I would say that Buddhist ethics supports more of a non-abolitionist research stance. So this is not to say that we should completely stop all research on invertebrate organisms, or even that we should extend more regulation to them. Rather that we should just think about these things more from alternative perspectives. So overall conclusions across both chapters. Firstly, comprehensive research on the potential non-targets of these Paleoditis biocontrol nematodes is needed and necessary in understanding how they might impact our ecosystems here. And then secondly, exploring diverse frameworks is really valuable in providing or in pursuing more ethical and compassionate research. And I think it's a worthwhile, whether it's Buddhist philosophy or not, just sort of thinking about alternative ways of conceptualizing your research, I think can be really valuable. Okay, and so with that, I want to get into some acknowledgements. I have notes because I don't want to forget anybody. But firstly, I would like to thank my wonderful committee members, Dr. Rory McDonald, Dr. Kirsten Grover-Colvert, and Dr. Yang Ming Di. I am really grateful for your support, and your guidance has been really invaluable to me throughout my program. I'm really grateful to you. Thank you. I also wanted to thank my funding sources through USDA, as well as my collaborators, Dr. Casey Richard, Dana Howe, and Indira Kulkarni. Thank you for all the knowledge and help you've shared with me. I feel extremely lucky to have spent so many hours in the field and in the lab with you. And especially, I'm so lucky to have started my program with Indira as well. I also wanted to give a special thank you to Dr. Jeffrey Barstow for supporting my philosophical exploration. Huge thank you to the IB office staff, Tracy, Teresa, Tara, Melissa, and Rowan. Your kindness and support throughout all of this has also been instrumental in completing my program. And I'm also incredibly grateful to my lab members and colleagues, Jasly, Brenna, Deanna, Ritu, Ali, Isabel, Grace, Kim, Subi, and Kayleen. And of course, a very heartfelt thank you to my advisor, Dr. D. Denver, for believing in me and giving me this opportunity to be part of such an amazing team and an amazing community here. I also wanted to thank my family for their constant love and support. And finally, thank you to my friends and loved ones, as well as the IB department, all of my students, collaborators in Nepal, and just everyone in the community for being so kind and welcoming to me when I came here. This experience has been incredible, and I've grown in so many ways, so thank you.
That was awesome. Thank you, Emily. Both again.
Okay, thanks everybody. Thank you, Emily. So no questions now from the committee members, but if anyone else, in the audience would like to ask a question, that's it. I'll have a chance. OMG.
Fantastic talk. Love it. Like, I think we're so easily to combine those two topics. I have a question, not helpful on the other side of things, say that, but the research side, really interested in that PC adult treatment. Like, that was such a drastic difference, like almost measurable. Are you aware of how they might have changed the NEMA slug or whatever it's called, like the treatment from that pH to the PC? What they changed in the formula, what they changed, how they slug, because that's a really drastic.
Yeah, so I guess one thing that is important to note is that the that we used for the pH nematode is closer to the real nematode formulation than the strain of P. californica. Is not pure hermaphrodite. So we're not why there has been literature to show that PC is not as effective in other slug systems. So yeah, it's a really interesting result. And looking at the two individuals that died, I do believe that they died due to nematode infection, but somehow it didn't. It wasn't affected to the other slugs in the container, which is a really interesting result. Yeah.
Yes, thank you.
Side note.
Oh, okay. One beautiful press did a really amazing. I'm also interested in the C adults, and I guess you said like suffering can't be quantified, and your experimental design was very about like the real like mortality cost, right, of the NEMA slug like nematodes. Is there a way that I guess quantify some people effects on the physiology of the slugs, and like what sort of like ways, like did the slugs that were like not dead look completely normal, or did they also suffer in like maybe a shade of gray?
Yes, so that's a great question. So yes, sometimes, oh, there we go. Some lethal effects are something that is studied in other systems, and I think the major one, especially for nematolite applications, is inhibition of feeding. So sometimes all that farmers actually do care about is whether the slugs will stop eating their crops. And so that is something that I noticed in this experiment with the banana slugs as well. And moreover, they had a lot of physical characteristics of infection, like you can see the slide on this one. There's a dead one at the back, that top right image. They would have this like really crazy slime generation. It seemed like maybe in an attempt to shed the nematodes off the back body, but there were definitely physical signs, discoloration, and also sometimes, in addition to not feeding, their sort of like mouthpiece would be extended more than usual.
So even in the ones that didn't die, the Nema slug product could be successful by its own metrics, and therefore devastating to the non-target species. Yes.
Post Lincoln.
Me?
Yeah.
Great job, Emily. That was a fantastic presentation. I'm curious, you were talking about like how to limit harm in vertebrates, and at the end of the experiment, you euthanized any surviving slugs. Did you consider maybe releasing any of the healthy or like control slugs back into the wild at the end of the experiment? I know in vertebrate research, we're often not allowed to do that, but it sounds like there are no like guidelines in invertebrate research saying you can or can't do that. So that's something you considered.
Yes. Let's see if I can get that. Oh, this image actually. So here, this actually is me releasing the control groups back into the environment. And that was also sort of an ethical question. We did bring them into the lab, and we're not entirely sure how to quantify how that changed them individually. And so I did end up releasing them back into the wild, and it felt really good. It felt like much better than the ones that I killed. So, but yeah, and I was only able to do that because there are no regulations around invertebrate organisms.
Did you say that you got all of your slugs from one location in Newport?
Yes.
How did you figure out what area you were going to take your slugs from, and did you consider like how that might affect the ecology of that area, taking so many slugs from their natural habitat?
Yes, that was definitely a concern I had going into the experiment. And so we targeted an area, thanks to the expertise of Dr. Casey Richard, an area called Bay Creek and I think Forest Grove Park. They're sort of two parts of the same park separated by a hill, but they have these really massive skunk cabbage patches. And so banana slugs are actually not an at-risk group themselves in terms of endangered species or anything. They are very prevalent here, and there are tons of them, and we came across tons of them. So I felt like I went in with enough confidence to know that removing so many would not be a major hit to the ecosystem. But that is only what I guess based on like others' expertise and past knowledge. So that was also sort of, I guess, on my part. A good question.
So when you started your master's, did you plan to have both chapters, or did the second chapter kind of evolve out of, practice of the first?
Yeah, that's a great question. I did not expect to have this as my second chapter. In fact, I thought that the topic of biocontrol itself is already such an ethical sort of discussion. I really expected to focus in on the biocontrol topic in particular. And then the moment where I had to autoclave the slugs really solidified for me that I wanted to explore it a little bit more.
I'm thinking about your results in like a larger ecological context. Are there things that, for instance, might eat or prey on the nematode, and how might you think like that, if for instance we did start using the Nema slug product in the US, how essentially the like prey interactions between things that are eating the nematode or things that are eating the slug or things like how to affect that to either control or not control the infant population, like if it were to spread, does that make sense?
Yeah, that's an interesting one. So I do know in other nematode systems, they often have like multiple intermediate hosts before reaching their target. I'm not sure, it's not a part of the Paleogenesis nematode life stage, but they can be pretty hardy in certain circumstances and they can't live off bacteria alone. So I think that would be a really interesting question. Potentially dispersal of the nematodes to further areas would be a product or a consequence of that, but that's
Hello, Emily. Going back to your first slide about mortality, the extinction rates of mollusks, you don't have to go back, just overall, is there any evidence that some of those extinctions were caused by their own parasitic nematodes? Like, were mollusks dying from that or from other causes like desiccation? I'm wondering about the realism of nemoslug spreading and actually decimating things in nature versus the very contrived used in a plantation where very high density food, very high density of slugs, and therefore it works there, it's spread fast there, but can they actually infect widespread slugs?
Yeah, that's a great question. So, a lot of that statistic of terrestrial gastropod extinctions is made up of native snails of specifically Pacific Hawaiian Islands. So, and that is resulting from failed biocontrol attempts. So, in terrestrial gastropods, because they are commonly pests to crops and gardens, there have been a lot of prior instances of failed biocontrol attempts, oftentimes in the form of other insects, not necessarily nematodes, I don't think, but particularly there was a predatory snail that was introduced to the Hawaiian Islands and other islands in the region that just totally decimated a bunch of native species as opposed to even affecting the pest populations that much. And so as for nematodes, I think that is more of a question to be explored, given that they are so small and particular to their host in certain instances, but we do know that biocontrol, introducing species in a form of biocontrol has been absolutely devastating to the terrestrial gastropod group in the past. And so, yeah, it's interesting to explore how they actually would come into contact with nematodes. And so I think it would be really cool to do like a mesocosm experiment where you're sort of having a more natural area and seeing if they really would come across that soil.
Thank you.
Left too much time, Emily, might be.
I was curious, what's known about how the nematodes actually killed the slug?
Oh yeah, so I didn't really talk about that. It seems that the nematodes themselves have a host of a bacterial community on their body. And so when they move inside the cavity of the slug, that bacteria sort of spreads out and does its own level of decomposing and infecting the body. And then the slugs are just there to sort of feed off of it, the bacteria that is generated from that.
Do you know anything about the bugs, like immune system and how they might be? This one is particularly, you know,
Yeah, it's really unknown, especially in slug systems. A lot of the research is concentrated on agricultural pest management. And so there has been interesting research, particularly in snails. And I think my colleague Deanna will be looking into this more, but snails have been known to trap nematodes in the inside of their shell a little bit when they're forming new calcium deposits. And so that is thought to be a defense, like an immune system mechanism. I think observationally what I saw is a lot of like trying to capture them in the slime somehow, and then shed them off. But I think given the limitations of my experiment where they were kept in an enclosed container was not very effective from what I saw. But yeah, not as much as known about that.
Last one, go ahead.
Okay, so you've mentioned, and we've talked about this, in various contexts, how we're not sure if the nemoslug species or strains are native to, the United States and the Northwest, and is that a problem? Do we have any research on the effect of nematodes on native slugs that we know are native?
Oh, well, I would say the banana slug is actually native, not just to Oregon.
No, I mean the nematode if the nematode is
Oh,
native.
I see what you mean. No, not yet, but I do, okay. That's awesome because now I can go to my secret slide.
Oh my god, how'd I know?
Geo, I think I messed with you.
Okay.
Okay. So, very interesting question. We actually do have research and evidence, actually carried out by Dana Howe in the front here that, the Hermaphrodita, the species, the lineage that is used in the Nemslug product has made its way over to the US. And so that's something I didn't really touch on because it's sort of its whole own project by itself. But this nematode does seem to have made it over here into US soil, probably just by accident, by like plants coming over from overseas or something like that. But interestingly, we also, like in my first few months of grad school, we were doing poop surveys of banana slugs. So you can collect their poop, put it on a petri dish, and see what nematodes come out of it. And so we actually found a different strain of P. californica that seems to be potentially native here. And in the introductory BI 221 laboratory, we've actually had students collect different like poop samples from slugs, and we found that same P. californica strain in a few of them. So it does seem to suggest that there is an infective nematode that is native to this area. And I did like a really small pilot experiment using that particular nematode, and it did kill the gray field slug in like a smaller scale design. So that is the interesting lead that we are wanting to expand.
Do you think the potential, the potential possibility that P. californica is native could play a role in the difference in your results between P. california and Hermaphrodita?
Yeah, absolutely. I mean, the strain in question that I isolated was not used in the experiment. But the one that we have used was isolated from other dead slugs. So that is definitely a factor in not being sure like which one is more effective or which strain was more close to the dead slug formulation. That's what it's.
All right, we're gonna wrap it up. Great question. There's the final one, gratitude.